Let us start the video with a little information. With the most advanced photographic cameras that science has ever discovered, can you guess the highest megapixel image we can film with those? Maybe with a DSLR we can take up to 50 to 60 megapixel image, or the best scientific cameras can take up to 250 to 70 megapixel images. But the human eyes are even better than those cameras in terms of megapixels in an image. The resolution of human eye is 576 megapixels. You know, that's huge. Ever since I knew this information, a question has been spinning in my mind. Even though we have such a powerful optical organ, why do we need a telescope to look at things far away from us or a microscope to view small things? Hello and welcome. In today's video, we will try to understand why we need a microscope to view small objects. We will never be able to make out the answer until we know the structure of the eye. Our eye is perhaps the most interesting of all optical instruments. Eye has three main layers. The inner layer is known as the retina, the middle layer is called the uvea, and the outer layer, we call it the fibres tunic. The outer layer consists of the sclera and the cornea, which are continuous with each other. The sclera is the white part of the eye, and the cornea is the clear, transparent dome-like structure positioned at the front of the eye. Uvea is the vascular middle layer, lies just beneath the sclera. It is composed of three main parts, from front to the back: the iris, the ciliary body, and the choroid. Iris is the anterior-most part of the uvea. There is an opening at the center of the iris known as the pupil. It gives color to the eye and acts like a diaphragm of a camera. So, it is responsible for controlling the diameter and size of the pupil and thus the amount of light reaching through it to the lens. Second part of uvea is the ciliary body, which is a circular structure and extension of the iris. The eye lens remains suspended from it, and by applying pressure of the ciliary muscle it can change the shape of the lens necessary for focusing light coming from a near object. The process is called accommodation. The posterior portion of uvea is choroid, sandwiched between the sclera and the innermost layer, retina. It is the vascular layer of the eye and provides oxygen and nourishment to the outer layer of retina. The lens of our eye has an ellipsoid biconvex shape. It is a transparent crystalline lens that bends the light rays for the second time after the cornea. The lens will direct the light rays towards the back of the eye and ultimately focus those rays onto the retina to form an inverted image of the object. Retina is the innermost layer. It is a light sensitive layer composed of several layers, including one that contains specialized cells called photoreceptors, the cones and the rods. The rod and cone cells act as transducers. Do you know what a transducer is? Transducers are devices that convert energy from one form to another. So the rods and cones act as biotransducers that converts the energy of the light, that is photons, into electrical signals. Those electrical signals will travel from retina through the optic nerve. Optic nerve is the cranial nerve too that transmits visual information from the retina to the occipital lobe of the brain. The brain then interprets the signal to create visual perception or into images that we see through our eyes. In short, that was the structure of the human eye. We will discuss the detailed structure of the eye in another video series. Now let's see the light path of our eye. The eye captures the image and then sends it to the brain by optic nerve. As light bounces off an object or is coming off a source from in front of the eye and comes into our eye through the dome-shaped window of the eye, that is the cornea. The cornea bend those rays for the first time and will help direct the light towards the pupil. Once the light comes past the pupil, it will go through the eye lens. So, that was the simplest path of the light entering our eyes. The eyes, like any optical instrument, has some limitations. Now it's time to find out why we with our naked eyes are unable to view very small microscopic things. From the common sense of using a camera, we can say that if we are to capture an enlarged image of an object, we need to zoom the lens. That is, we have to move the lens towards the object. The same thing cannot be done with our eye lens. We cannot shift it. As we know that our lens remains suspended in place by suspensory ligaments known as zonule fibres. These ligaments attaches to the lens at its equator and connects it to the ciliary body. So it is impossible for us to zoom our lens to create an enlarged image of an object. There is another way to have the zooming effect in image without shifting the lens. Yes, by shifting the screen away from the lens where the image will form. In case of our eye, the screen is the retina. Can we move it forward or backward as per our will? No, it is also not possible. The distance between the lens and the retina is fixed. The distance is also known as the focal length of our lens, and it varies from man to man from 1.7 centimeter to 2.2 centimeter. What does it mean? It means that our lens is bound to focus the light rays coming from the object on the retina, which is located just 1.7 centimeter behind it. The third limitation that I am about to tell you is another major obstacle to view small object, and it is the so-called near point, beyond which the eye cannot focus. We know that if we bring something close to the eye, it looks a little bigger. But is there any limit to how much something can be brought closer? A person with normal vision can see objects clearly at a distance ranging from 25 centimeter to infinity. What does it mean? It means that 25 centimeter is the nearest point from the lens at which an object can be placed, and we can still have a sharp image produced on the retina. If we place an object too close to our eye, say for instance within 23 centimeter from our eye, our lens will not be able to focus the light rays onto the retina, and this will cause the image to look blurry. By doing a simple test, let us try to understand the matter. I request you to keep an eye on the brand name of the pen in my hand and watch carefully. As I gently move my hand towards the camera, there will be a time when you will not be able to see the text on it. It means that the pen has crossed the near point of your eye. 25 centimeter in case of your eye or our eye is known as the least distance of distinct vision, and we cannot focus beyond the near point. Now, if we combine all those three limitations that I have already taught you, what is the thing that comes out? I hope is now clear to you that neither we can move the lens towards the object, nor we can shift the retina backward to move any object to look large. Furthermore, it is also not possible to move close to the object beyond 25 centimeter to have an enlarged view of the object. The next two limitations that I am about to tell you cause even bigger problems in making little things look bigger. And the first one of them is the magnification power. Magnification power is the ability of a lens to increase size of optical image over the size of the object. So for example, say this is our tiny object or specimen, which is so small that we cannot view this with our naked eye. So what do? We will use a lens. As a reflected light rays from the object pass through the lens and a large image is made on the other side. So this is what we see. Image is bigger than the actual size of the specimen and it is magnified. More powerful lens will have more magnification. Magnification is given by the equation m is negative L prime by L, where negative L prime is the distance from the lens to the image. In case of human beings, it is the distance from our lens to the retina, and it always remains the same, a little over 1.7 centimeter. And L is the distance between a lens and an object, which is 25 centimeter in case of human beings. As we see objects in its greatest magnified form when the object is placed at the near point, that is at 25 centimeter. So the maximum magnification that could be achieved by our eye is 0.068. Generally, this is defined as 1x magnification. And it turns out that we cannot magnify any object with our naked eye. Out of many limitations of our eye, the last one that I will be discussing today is the limited resolving power of our eye. Resolving power is the ability of a lens to distinguish between two adjacent objects as discrete. Actually, resolution is the shortest distance between two points that can be distinguished by an optic system. The less the distance that can be resolved as separate entities, the more the resolving power of the lens. Let's suppose this is the image of an object we are viewing with our naked eye. And now, if I place a magnifying glass over the same object, and by doing this, if the object appears as separate entities, we can say that the resolving power of the magnifying lens is higher than our naked eyes. Hope this makes sense to you. The resolution of a typical unaided human eye is about 200 micrometer, whereas eukaryotic cells are roughly 10 micrometer to 100 micrometer in size. And I would like to give you an idea about the prokaryotes like bacteria. Eukaryotic cells are about 10 times the size of a prokaryote, roughly 0.1 to 5 micrometer in diameter. So, how is it possible to view such microorganisms or cells with our naked eye, having a poor resolving power of 200 micrometer? Apart from those, it has some other limitations too, like due to its small size, it has got a limited light gathering power, as well as it cannot accumulate light and store image for future references. The most significant drawback, I think, is its limitation of viewing only in a visible spectrum of electromagnetic radiation. Using a microscope decreases the resolution to distances as short as 0.2 micrometer, because a microscope is an instrument used to see objects that are too small to be seen by the naked eyes. So that's the end of the video. Hope you liked it. I would ask you to do subscribe my channel and encourage me. Thank you.